First, we will look on to the overview of glycolysis. In glycolysis, a molecule of glucose is degraded to give pyruvate, which is a 3-carbon compound, in a series of enzyme catalyzed reactions. Glycolysis is also known as EMP pathway, that is Embden-Meyerhof-Pernas pathway. Free energy released from glucose is conserved in the form of ATP and NADH. Glycolysis consists of 10 steps. The first 5 steps is known as the preparatory phase, whereas the last 5 steps constitute the payoff phase. Now we will study each step in detail. The first step is phosphorylation of glucose. As we can see in the image, glucose is converted to glucose 6-phosphate. The phosphate group attached to glucose 6-phosphate is obtained from ATP, which is then converted to ADP due to loss of one phosphate group. This reaction is an irreversible reaction and it is catalyzed by the enzyme hexokinase. Hexokinase requires Mg2+ as a cofactor. This reaction is known as phosphorylation of glucose. The second step is conversion of glucose 6-phosphate to fructose 6-phosphate. Glucose 6-phosphate is an aldose which is converted to fructose 6-phosphate, which is a ketose. This reaction is a reversible reaction and it is carried out in the presence of phosphohexose isomerase. This enzyme also requires Mg2+ as a cofactor. The third step is phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. In the image we can see that fructose 1,6-bisphosphate again gets one more phosphate group from ATP, which is then again converted to ADP. This reaction is an irreversible reaction carried out in the presence of phosphofructokinase 1, which also requires Mg2+ as the cofactor. Phosphofructokinase 1 is an allosteric enzyme. Allosteric enzyme means it has two sites, one active site and one regulatory site. This reaction is known as the committed step. That means fructose 1,6-bisphosphate, when it is formed, it is confirmed that it will complete glycolysis pathway. The fourth step is cleavage of fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate, which is a 6-carbon compound, is converted to give us two products of 3-carbon compound each. One is dihydroxyacetone phosphate and other is glyceraldehyde 3-phosphate. This reaction is a reversible reaction carried out in the presence of aldolase enzyme. It is also known as aldol condensation. The fifth step is interconversion of the triose phosphates. The two products obtained in the previous reaction, in the previous step, is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. But only glyceraldehyde 3-phosphate can be degraded in the subsequent steps of glycolysis. So dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. So now we have two molecules of glyceraldehyde 3-phosphate. This reaction is a reversible reaction carried out in the presence of triose phosphate isomerase. This reaction completes the preparatory phase of glycolysis. Now begins the payoff phase with the step 6, that is oxidation of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. In the image we can see that glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate, that is one more phosphate group is attached to 1,3-bisphosphoglycerate, which is obtained from the inorganic phosphate. Here NAD+ is converted to NADH, which can give us ATP, as we know that 1 NADH is equal to 2.5 ATP. So this is the first energy conserving reaction that leads to the formation of ATP. This reaction is a reversible reaction carried out in the presence of enzyme glyceraldehyde 3-phosphate dehydrogenase. The product, that is 1,3-bisphosphoglycerate, has a very high standard free energy. The seventh step is phosphoryl transfer from 1,3-bisphosphoglycerate to ADP. 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. The phosphate group attached to 1,3-bisphosphoglycerate is taken up by ADP to give us ATP. This reaction is known as substrate level phosphorylation, means formation of ATP by phosphoryl group transfer from a substrate such as 1,3-bisphosphoglycerate is known as substrate level phosphorylation. This reaction is reversible reaction and it is carried out in the presence of phosphoglycerate kinase enzyme, which again requires Mg2+ as a cofactor. The eighth step is conversion of 3-phosphoglycerate to 2-phosphoglycerate. The phosphate group is shifted from carbon 3 to carbon 2 to give us 2-phosphoglycerate. This reaction is a reversible reaction and it is catalyzed by the enzyme phosphoglycerate mutase, which requires Mg2+ as a cofactor. The ninth step is dehydration of 2-phosphoglycerate to phosphoenol pyruvate. As we can understand from the word, dehydration means loss of water. So from 2-phosphoglycerate, one molecule of water is lost to give us phosphoenol pyruvate. This reaction is carried out in the presence of enolase enzyme. The product, that is phosphoenol pyruvate, has high phosphoryl group transfer potential, that is delta G naught is equal to minus 61.9 kilojoule per mole, while that of the 2-phosphoglycerate is only minus 17.6 kilojoule per mole. This reaction is also stabilized by Mg2+. The 10th step, that is the last step of glycolysis pathway and also the last step of the payoff phase. This is transfer of phosphoryl group from phosphoenol pyruvate to ADP. The phosphate group from phosphoenol pyruvate is again taken up by ADP to give us ATP and product, that is pyruvate. This reaction is again known as substrate level phosphorylation, as we get ATP by the transfer of phosphoryl group. This reaction is a irreversible reaction carried out in the presence of pyruvate kinase enzyme, which requires K+ or either Mg2+ or Mn2+. The pyruvate first appears in its enol form, then it tautomerizes rapidly to its keto form, which is more stable. So therefore, it has a large negative standard free energy change. So this completes the glycolysis pathway. Now we will see the net gain of ATP. 2 molecules of ADP and 2 of inorganic phosphate are converted to 2 molecules of ATP. And 2 hydride ions are transferred from 2 molecules of glyceraldehyde 3-phosphate to 2 of NAD+, giving 2 NADH. So 1 NADH is equal to 2.5 ATP. So 2 NADH will give us 5 ATP. So 5 ATP from NADH and 2 ATP will give us a total of 7 ATP. This completes the glycolysis pathway. Thank you.